I'm going to talk about how we can use mutations to try and identify cancer type. And so, as we've just heard, cancer is a process of somatic evolution. And one of the most important events in the evolution of a tumor is metastasis. When a tumor metastasizes, some cells from the primary tumor gain the ability to migrate to and colonize some other organs within the body. And metastasis is a major clinical challenge and is the primary cause of cancer-related mortality. Because cancer is an evolutionary process, metastases contain some ancestral information about the primary tumor in the form of mutations that are common amongst the cells in the metastasis and the cells from the primary tumor that gave rise to the metastasis. They typically contain some additional mutations and a large amount of phenotypic divergence from the primary tumor. And so most of the time when a patient comes into the clinic and has metastatic cancer, it's obvious what the primary tumor is. But in about 3 to 5% of new cancer diagnoses, a patient will come in with metastatic cancer and no obvious primary. And this constitutes a carcinoma of unknown primary and is about 50,000 new diagnoses per year in North America. Because tumors' tissue of origin and histopathology are some of the strongest determinants of its clinical behavior, it's important to try and figure out what the primary tumor is for these patients. Unfortunately, when studies try and examine the ability for pathologists to identify primary tumor site, they find that pathologists are typically unable to do so accurately. And so given this obvious clinical need, it's natural to ask if we can use genomics to try and improve the ability to identify primary tumor site. And the obvious place to start is to try and look at driver mutations or mutations to cancer genes. And so just here I'm showing the kind of classic model for the development of pancreatic adenocarcinomas. And you can see that pancreatic cancer typically has mutations to these 4 well-characterized cancer genes. And so you can imagine that if a patient comes into a clinic with an unidentifiable cancer and you see these 4 mutations, you may have some reason to believe that they have pancreatic cancer. And so based off of this intuition, my colleague Wei Zhao at the Ontario Institute for Cancer Research trained a series of classifiers to try and identify cancer type based off of mutations to driver genes and based off of alterations to driver pathways. And what we found was that typically these features were unable to accurately identify cancer type and on average had F1 scores of less than 0.5, which is actually worse than the pathologists I mentioned on the previous slide. And so at its core, the problem of trying to identify cancer type is actually a problem of trying to identify cell type. And one of the defining features of a cell type is its chromatin state. And at a very, very, very high level, chromatin just describes how DNA is packaged within a cell. And you can broadly split chromatin into regions that are open or accessible and regions that are closed or inaccessible. And so if there was a way to assay Metastasis's chromatin state, you could probably train some sort of machine learning model to identify cancer type. Unfortunately, chromatin tends to be a little more transient than something like DNA, and it's not obvious that the chromatin state of a metastatic tumor is going to match the chromatin state of the primary tumor. Fortunately, we may be able to use mutations, or at least mutation density, as a proxy for chromatin state. And so this figure, the x-axis is the chromosomal coordinates. The blue line represents the degree of closed chromatin in a region, and the black line represents the number of C to T mutations in these melanoma samples in TCGA. And so the really striking thing about this figure is that there's a super high concordance between the blue and black lines. Wherever you tend to see an increase in closed chromatin, you see an increase in mutation density. And this is probably due to inefficiencies in DNA damage response. And because mutations contain ancestral information in the sense that mutations from the primary tumor are often going to be found in the metastasis, mutations can provide us with information about the primary tumor, and they allow us to partially encode the chromatin state of a tumor based solely on its mutations. We can gain additional information about cell type from looking at the different mutational processes that are active within a tumor. And so people typically do this by constructing mutation signatures, which consider short-range sequence context around single nucleotide variants. And so there are a number of different mutational signatures, and some of them have a large amount of cell type specificity. Good example is the smoking signature, or signature 4. If you have an unidentified tumor and it has a large amount of exposure to mutation signature 4, you probably have some reason to believe that it might be a lung cancer. And so if we return back to our original problem, what this project aimed to do was to train a series of deep neural networks based on primarily patterns of somatic pasture mutations to accurately discriminate between multiple cancer types. To train this model, I used data from the pan-cancer analysis of whole genomes, or PCOG. And PCOG has collected whole genome sequencing of about 2,800 cancer samples from 39 different cancer types. And for the purposes of this work, I limited myself to tumors that had, or cancer types that had at least 36 unique donors in PCOG, which left about 2,600 samples from 24 different cancer types. I had to do a little bit of work to actually derive features from pasture mutations. And so this was done in two ways. First is what I refer to as the mutation distribution. And this corresponds to the relationship between mutation density and chromatin state. And so what we do to construct these features is we basically take all the autosomes in the genome, we glue them together head to tail, and split them into 1 megabase regions. And in each region, we simply count the number of single nucleotide variants and throw out all other information about those mutations. And so functionally, for the purposes of this model, each mutation is equivalent. I also include information about the different mutational processes active within a tumor by using the counts of the different mutation types. And so using these features, I trained a number of different classifiers, allowing me to do two things. First, identify the best overall performing model, but also to compare the ability of different features derived from mutations for identifying cancer type. And so here I'm just showing performance for 3 of the models. F1 scores on the y-axis. The individual data points represent the F1 score for each of the 24 cancer types for a given model. And so the first model was the best possible random forest model, and it was trained on the features that I described on the previous slide. Then we have a neural network also trained on those features. And we trained an additional model using the features described on the previous slide, but we also added explicit information about which cancer genes or cancer pathways were altered. And the main takeaways here are that the neural network that was trained primarily on features from pasture mutations was the overall best performing model with an F1 score of over 0.92, suggesting that pasture mutations are sufficient for accurately identifying cancer type. And we kind of had this interesting side result that when we added explicit information about which cancer genes are altered, we saw a slight decrease in overall performance. And this is probably due to a small degree of overfitting. And so here I just have a confusion matrix for the best performing model. And we have the model's predictions over here on the x-axis and the true cancer type on the y-axis. And this is averaged over an ensemble of 10 different neural networks. And so you can see that overall accuracy was about 91%, which is roughly double the pathologist that I mentioned on the second slide. And 20 of the 24 cancer types could be identified with an F1 score of at least 0.8. When the model makes mistakes, they're often explainable due to common cell types of origin or other biological features. A really good example of this are the misclassifications between stomach cancer and esophageal cancer. These are tumors that arise from very similar cell types. There's a small number of misdiagnoses due to tumors that actually occur in the gastrointestinal junction. And they have very similar patterns of mutational exposures throughout development. We see something similar when we look at misclassifications between the two B-cell malignancies. So these cancers arise also from the same cell type of origin, just at different developmental stages, and also have very similar patterns of mutational exposures. And what we can see is that while the model does a very good job of differentiating these two cancers from all the other cancer types, it often mistakes one of them for the other. Despite these difficulties, the classifier does quite well on some highly related cancer types. A good example is glioblastoma and piloastrocytoma. These two cancers also have similar cell types of origin, but the classifier almost never mistakes one for the other. And we see similar results when we look at kidney chromophobe carcinoma, kidney renal cell carcinoma, and a number of other highly related cancer types. And so, yeah, these results are very good, but as I mentioned, we're motivating this model as something to try and identify the primary tumor type for metastases. And so it's important that I actually evaluate the model on data from metastases. And so we are fortunate to receive whole genome sequencing of about 2,000 metastatic tumors from the Hartwig Medical Foundation, and an additional 200 metastatic pancreatic adenocarcinomas from Steve Gallinger at the Ontario Institute for Cancer Research. And despite the fact that these tumors were biopsied in a different manner, had different sequencing protocols, different analysis pipelines, and that many of the cancers were sequenced following treatment, our classifier that was trained solely on data from primary tumors was able to accurately identify cancer type with an overall accuracy of 83%. And it does quite well on any cancer with at least 100 examples, suggesting that some but not all of the drop in performance is due to some sampling bias. So just to quickly summarize, what I presented here is a series of neural networks that are trained almost entirely on patterns of somatic pathway mutations, which can accurately discriminate between 24 common cancer types. The model has immediate clinical applicability in identifying the primary tumor for carcinomas of unknown primary, and we're exploring different manners of moving it into a clinical setting. And we have this kind of really neat result that the classifier that was trained solely on passenger mutations was the best performing model, suggesting that at the very least, there's enough information in passenger mutations to accurately identify cell type. And in addition, when we actually add information about which cancer genes are altered, we see a slight drop in overall performance. With that, I just want to thank. So,
we are having temporary problems with our service passenger elevators. Please, all employees, use the service elevators for the stairs. We are temporarily having problems with this passenger elevators. Please, all employees, use stairs or use the service elevators. Thank you.
Right. So I just want to thank my supervisors, Quaid and Gary, everyone in the Morris Lab, my collaborators on this project, especially Wei and Paz, and Jacob Mora, who's a super talented undergraduate student who's been working on me with some stuff that I didn't get a chance to get into today, and the OICR and Vector Institute for supporting this work.
A question regarding the confusion matrix. I think there's explanations of why the model sometimes fails. Make a lot of sense to me. But what about the frequency of the tumor entities in the training data?
I'm sorry?
What about the frequency of the tumor entities in the training data? I would assume that some entities are way more represented and give more examples with patients than with others. So would that also influence the performance of the model?
Okay. Yeah, so sorry. So here I just have the size of the example, sorry, the cancer type on the x-axis and the F1 score on the y-axis. And so, well, if you go over about 150 examples, it does reasonably well. We see kind of a large amount of variability for cancer types that have fewer than 100 samples. You have some that probably have something very unique, like melanoma, where you're going to see a lot of UV exposure. So that might be one of the reasons it performs quite well. And then you have some cancer types like stomach that do quite poorly, but I suspect that much of that is due to this relationship with esophageal cancer. And if we had more stomach cancer samples, we might be able to tease out the differences between those two cancer types much better.
Clear. I was just wondering why your driver line mutations aren't. Is it because of the diversity between the drivers across all cancers?
So what I suspect is that there's a relatively small number of highly mutated cancer genes that are relatively common. And then those kind of cancer type specific driver mutations are really only specific to like 20%. So like something like KDM6 is like often mutated in pancreatic cancer, but it's like 30% of pancreatic cancers, which isn't really enough to accurately identify a cancer type. And if you group them into pathways, you find that they're basically just the pathways for like the hallmarks of cancer. And so those are going to be mutated in every tumor.
Yeah, that's not. You said that you use whole-genome sequencing.
Yeah.
Have you also tried it just with the exome?
I'm working on it. It's not working very well right now, but it's a work in progress.
Just for curiosity, because it would decrease the cost of it.
Yeah.
The last question. How does the copy number operation or aneuploidy affects the mutation frequencies both in the chromosomes and within the regions, and has that any impact on the
So kind of when we were doing internal validation, there's a point where we did explicitly included the copy number state as a separate feature, and it didn't improve performance at all. There's a point where we tried to correct the mutation counts for the background copy number state, but that also didn't improve performance.
Attention, attention. This is your fire safety director speaking. The elevators are back in service. All the elevators are back in service. Thank you.
But kind of implicitly, copy number state is included in the model because if you have an amplification or deletion, it'll affect the expected number of mutations that you're going to observe in any given region of the genome. So while it's not explicitly included, just looking at the mutation counts has some information about what the background copy number state is in a given region.